Reproduction 101. A couple of terms that you should know include mitosis, meiosis, gametes, and zygote. Okay, let's start with mitosis. What's the definition of mitosis? Let me try to bring the chat up so let's see in case anyone has a question. Okay, so what is mitosis?
It's to copy, right?
So mitosis is a form of cell division. And it's a form of cell division where it occurs with what type of cells?
A somatic cell.
Yeah,
Zygotes.
somatic cells. What do somatic cells mean? And you don't have to have the exact definition, just describe what a somatic cell is. How can you tell it's a somatic cell?
Cells that are not part of the sex reproductive system?
Yeah, so it's all cells except gametes. Any other way that you can distinguish somatic cells?
Is it the number of chromosomes?
Yeah, they tend to always have how many?
46?
Yes. And 46 is one way of saying it. What's another way of saying it?
Diploid.
Yep. And what's another way of representing diploid?
2N.
Yes. Excellent. Okay. So that brings us to another term, chromosomes. What are chromosomes?
Aren't they made up of DNA?
Yeah. Okay. So chromosomes are tightly coiled up DNA. And what you'll find is that they can go into, for example, in humans, you'll have chromosome 1, and then you'll have another copy of it. Chromosome 2, and then you'll have another copy. 3, 3, all the way down to 23. Now, the 23rd pair can be either an X and an X or an X and a Y. Why is the 23rd pair different than the 1 through 22nd pair?
Because that differentiates the sex?
Yeah. So this would be the sex chromosome. The chromosome number 23 is a sex chromosome. It may be the same, X and X, or X and Y. Where chromosomes 1 through 22, because they're identical, they're called autosomes. Okay? Now, that's why you can say that altogether we have 46 chromosomes, or we have 2N, because we really do have copies of each other. And that's why you have the 2N. Okay? Although there's a little exception for the sex chromosome, especially if it's not the same. Okay? So that's mitosis. So mitosis is a cell division of somatic cells, and the somatic cells tend to have 46 chromosomes, or diploid or 2N. Where meiosis is also a form of cell division. Okay? And it is a cell division only for gametes. And it's essentially a way to decrease the number of chromosomes. Okay? So meiosis is a form of cell division, only occurs in gametes. It's a way to decrease the number of chromosomes. So from 46, it's going to end up being how many?
23.
Exactly. And that's why the 23 chromosomes are referred to as haploid, because it's half or just one end. Okay? Now, if females, in terms of gametes, sex cells, so in females, it would be an ova, an ovum, or an egg. And in males, it would be a sperm. And these will always have 23 chromosomes or one end. Okay? So if we were to bring two gametes together, it forms a zygote. So a zygote is the fertilization of an ova, an ovum, or an egg by a sperm. So when it comes together, the chromosomes combine. But essentially, a female can only contribute what?
The egg?
The female can only contribute the X. So it doesn't matter which egg she releases. In ovulation, it can only have one X. Where a male will have a sperm that can either be what or what?
X or Y.
Exactly. Okay? So a male can contribute either an X or a Y. And there's a 50% chance. Okay. Even though it's weird because in some families, like all they have is girls or all they have is boys. But statistically, it really is a 50% chance. Okay. So someone should have told Henry VIII this, right? When he killed all his wives because his wife only gave him daughters, because that's all they could give, because that's all they have is an X. So really it's his fault for not having this combination. Because it's the males that determine the gender of a child. Because they actually can contribute either an X or a Y. Okay? Questions on reproduction 101? Hopefully that clears up some definitions. Any questions before I move on to the female reproductive?
Can we just go into depth with meiosis later on in the folliculogenesis?
Oh, absolutely. Yes. And actually, meiosis is more discussed in oogenesis than follicular genesis. But yes, we will be going through that. That's usually what students have the most difficulty in. Yeah. All right. So that was a kind of a reproduction 101. So we're going to female reproductive system. In the female reproductive system, and a little bit, you want to make sure that you know the terms we just went through: gametes, fertilization, zygote, chromosomes, and cell division. So kind of just what we just reviewed. Okay? Be able to describe the functions of the female reproductive system, and then know the organs and their function. Okay? Which includes the ovary, the uterine tube, the uterus, the vagina, and the other external female organs. The one that we're going to spend time on, because we do have limited amount of time, we're going to spend some time on oogenesis, and then follicular genesis, and probably not in great detail, but I'll try to get into the menstruation cycle as well. Okay, so we're definitely for sure going to do the ovarian. and then maybe briefly talk about the menstrual cycle. All right. So anatomy of the female reproductive structure. So make sure you know it for both lecture and lab. And then for lecture, add on the function. Okay. So that brings us to oogenesis or oogenesis. You can pronounce it any way you want, but essentially it's the development of an ovum. Ovum is single, ova is plural, and then we use the term egg once it's released. Okay. Now, there's some very interesting facts about this process that distinguishes this process as female, where men go through a different process. Okay. And one of those big distinguishing factor is that a female develops the eggs that she has actually during her own fetal development. Okay. So when she is developing, she will actually develop all the eggs that she will have. So she'll be born with all the eggs that she will have in her lifetime. Unfortunately, that does mean what to the egg as the female ages?
The eggs also get older.
That's right. And that's why female who tend to be older, not 100% of the time, but there's a higher risk of what? If they get pregnant when they're older. Yep, miscarriage is one, and also chromosomal defects because the egg is just older. Okay, so that's one distinguishing factor is that a female is born with all the eggs she will have because she develops them during her own fetal development. Okay, so during a female's fetal development, she's developing other cells of in her body to form, for example, the brain, the heart. She's going to also form in her ovaries the eggs that she will have and she'll carry for the rest of her life. Okay. So during fetal development, she does start off with these stem cells called the oogonia or oogonia, whichever way you want to pronounce it. Okay. So the oogonia is the stem cell. Now the oogonia is currently 2N, which means what again? It has how many chromosomes?
Diploid?
Yep. So stem cell, diploid.
46.
Thank you. 46 total chromosomes. Okay. Now, during fetal development, she's going to try to make as many eggs as she will need in her lifetime. So this first part of oogenesis actually involves mitosis with the T rather than meiosis, because she's going to try to make as many as she can. Okay. So this first part is mitosis, and this happens during fetal development. Let me draw a line so that you can see the difference. So this yellow line right here represents what happens before she's even born. This is when the female is still in her mother's womb. Okay. So the process of oogenesis occurs, and it begins with mitosis. So in mitosis, what's going to happen? Someone describe mitosis for me.
It's when the cells like double, right?
Yeah, exactly. Yep. So what's going to happen, just like you said, it's going to copy. And then once it's done copying, it's going to divide. So mitosis copies and divides. So that means that temporarily, inside the cell, instead of 2N, when it copies, it's going to become what?
1.
No, copy first.
So two ends?
Two N's. That's a way of saying it. Yep. So 4N total.
4N.
Yep. So two N's, or you can say 4N, or a total of 90. Let's see, 40.
92.
Thank you. 92 chromosomes total. Okay. So there's just going to be a exact copy. And then, so that means in that cell, it's going to, after it starts mitosis, it's going to have 4N or 92, and then it's going to divide. So when it divides, it just splits in half. half of the information goes to one cell and the other half goes to this other cell right here. Okay? So when it splits, if you have one cell that has 4n and it divides into 2, how many n's should go into each one?
2.
Yeah. So this cell is going to have 2n and this cell is going to have 2n. Okay? Now, one of those cells stays back and continues to repeat this mitosis process. Okay? So one of those cells stays back and repeats this mitosis process, where the other one differentiates. You remember learning the term differentiates? What does differentiate mean again? It picks up a, yep, I'm sorry, go ahead.
Like changes into?
Yeah, so it picks up a specific function, so it changes a little bit, but it still is the same cell. It just changes a little in the fact that it picks up a specific function. So it's no longer a stem cell. It picks up a specific function. In other words, this cell has decided that instead of being a stem cell and repeating this process, I am now going to become an egg. Okay? So when that happens, it now is referred to as the primary oocyte. Okay? So the primary oocyte is what results from mitosis of an oogonia. One of them stays and repeats the process. The other one that's produced is now going to become an primary oocyte. Questions? Does that make sense?
Could you repeat that again?
What part?
The part where it copies and then it turns into a 2n, but then it goes back into mitosis or something like that?
Sure. So you start off with an oogonia, which is a stem cell, and that stem cell is going to undergo mitosis, which copies and divides. So at first it copies, so instead of going from 2n, it's now 4n, or 92 chromosomes total, and then it goes into the division part where it splits in half. When it splits in half, that new cell is going to repeat, one of the new cells is going to repeat the process and stays as an oogonia to undergo mitosis, where the other cell differentiates and becomes a primary oocyte.
Got it. Thank you.
Yeah. Okay. Now the primary oocyte is going to begin meiosis. In other words, because it's decided to become an egg, it's going to begin meiosis, but it stops. Why does it stop?
I saw a video that because we're born.
Yeah, exactly. But stops at birth. So it hasn't even finished meiosis one yet. It just begins it. But it stops at birth. Okay? So at birth, you essentially have just the primary oocyte. Okay? Now, it's going to start up again at what period of time in that girl's life?
Puberty.
Yes. So at puberty, this process continues. Why? What's released at puberty?
Hormones.
Yeah, what hormones?
The progesterone and the estrogen.
Not quite yet.
LH and FSH?
Yes. As a matter of fact, FSH first. So the
Yeah.
follicle stimulating hormone is what triggers this process every month at puberty. Okay? So the FSH is going to cause the primary oocyte to complete meiosis 1. Now, meiosis has two parts, meiosis 1 and meiosis 2. Okay, let's start with meiosis 1. I'm going to zoom in again. Okay. So let's start with meiosis 1 and give it a different color. So in meiosis 1, here you're just going to have a plain divide. The cell is just going to divide. And that's what happens in meiosis 1. So you can see here the primary oocyte starts to undergo meiosis 1, but it doesn't complete until puberty. At puberty, with the stimulus of follicle stimulating hormone, it's going to completely divide. And that's why from one cell that was a 2N, it's going to split and become 2 cells that is a what?
A haploid, or only one N.
Exactly. Because remember what we said about meiosis? Really, meiosis is a way to get rid of some chromosomes, right? So now this picture isn't quite accurate, okay? So after meiosis 1 occurs and it divides, it's actually going to divide into one cell that's large and has all of the cytoplasm, the cytosol, the organelles, and then that one N. The other one essentially is just a way to get rid of that extra chromosome. Okay, so the one that once it divides, one of them will keep everything, and the other one just spits out the chromosome. The one that spits out the chromosome is referred to as the polar body, and the polar body is going to go undergo a process called atresia. And essentially, atresia is where it just shrinks and dies. So essentially, it's a way of just to get rid of that. Okay. But that one that results, that keeps all of the good stuff, that's referred to as a secondary oocyte. We're just going to erase this right here. And we'll write it over here. Okay. So at the end of meiosis 1, you really only have one cell. From the primary oocyte, even though you produce 2 cells, one of them becomes a polar body and undergoes atresia. So really, the end result of meiosis 1 is one secondary oocyte. But now that secondary oocyte has how many chromosomes?
23.
Excellent. Exactly. Okay. So now this secondary oocyte just stays like this, and it will be ovulated. And what's ovulated? What's ovulation?
It's when the egg ruptures. No?
Not so much as the egg ruptures, but the follicle ruptures.
Mm-hmm.
And when the follicle ruptures, it does release the secondary oocyte. Okay? So the secondary oocyte is released upon ovulation. And once it's released, it's either going to be expelled if fertilization does not happen. It's just gonna be expelled, and then that's what ends up being released at the end of each month, called a process called menstruation. Okay. Now, if fertilization does happen, so if fertilization does happen, and here's the secondary oocyte. If fertilization does happen, the minute that sperm penetrates the wall of that egg is going to finish meiosis by undergoing meiosis 2. Okay, so meiosis 2 only happens if fertilization happens. Okay, so meiosis 2 only happens if fertilization happens. Otherwise, what's expelled is the secondary oocyte in menstruation. Okay, so if fertilization does happen, then that secondary oocyte is going to quickly finish meiosis 2. Now, in meiosis 2, you have a copy and divide. And this is what doesn't make sense to a lot of people, because you're like, it doesn't need to copy and divide. So why, but it's just the process that happens in nature with meiosis. So in meiosis, gametes just copy and divide in meiosis 2. So that's why, even though it doesn't really need to, it's going to. Okay? Because why? It has to finish meiosis 2 just by nature. Okay? Because technically, already the egg has one end, the sperm has one end, so technically that could be done, right? Yes, that is true. It could be done, but according to nature, it has to finish meiosis 2. So here, just briefly, that one end, okay, is going to copy itself. So temporarily, it's going to become 2N. Then it's going to divide, and it's going to kick out that extra N. And what stays in here is just that one N again. And the other end that gets kicked out is another polar body that essentially undergoes atresia and shrinks. Okay, so once it's fertilized, the minute the sperm penetrates that egg, it's going to finish off meiosis 2, not because it needs to, but because it has to, in terms of just finishing the process up. But essentially, it's going to, again, leave it with just one end. And then once meiosis 2 is completed, the N from the sperm will combine with the N of the egg. And that's what's going to form a zygote. And that zygote is going to return to being, going back to undergoing mitosis. Questions?
So when you say that we have to go under meiosis 2 and then we copy and divide the gametes, and then we finish meiosis 2, we end up with one sperm right there has one N and egg has one N, which becomes 2N?
Yep. And that's what a zygote is. It combines the chromosomes from the mom and the chromosomes from the dad.
Okay. And when you say that we have to kick off one N and go to the polar body, what do you mean by that?
So after meiosis 2, because in meiosis 2, it's going to copy. So one N becomes 2N, and then it's going to divide. So one of the ends stays in that egg, the other end gets kicked out and becomes a polar body.
Okay. Thank you.
Yeah, it's kind of confusing. It'll make more sense once we go through spermatogenesis, and then you're like, oh, yeah.
Thank you.
Yeah, but for now, know that in females, in the oogenesis, it's about quality. It's about producing the best one each month. And then, because that's not only the egg, that's not only just the ova, but that will eventually become the egg and the zygote, which, is the first cell of the first being. So it has to have all the good stuff in there. Because the sperm doesn't have anything. The only thing that the sperm has is just the chromosome from the dad. Okay? Questions? All right. So while that's, excuse me. While that's happening, what we didn't take into account is the follicle. So surrounding that ova, or that ovum, or that egg, are follicular cells. Okay? So there's follicular cells that surround that egg or that ova or that ovum, whichever way you want to say it. Okay? Now, starting with the oogonia, what surrounds the oogonia is what's referred to as the primordial follicle. The primordial follicle consists of follicular cells that surround the ova. Okay? So when the egg is developing under mitosis, so going from oogonia to primary oocyte, the follicle does not change. So there's no change in the follicle. Okay? The follicle only changes at puberty. So at puberty, that's when you start to see a change in the follicle that surrounds that ova. And the reason for that is because at puberty, what hormone is released?
FSH and LH
Start one first.
versus FSH, right?
Yep, yep. So FSH starts the whole story of that month. Okay. Now that primordial follicle, you should note that it is simple squamous epithelium. Okay, because the name changes as the structure changes. So what's surrounding the primary oocyte and actually the oogonia too is the primordial follicles, which are formed by the follicular cells. And in terms of tissue, those cells form the simple squamous epithelium. Okay. Now at puberty, FSH, and the reason why it's called follicle stimulating hormone is because it actually stimulates the change in the follicles more than it does in the egg itself. Okay, so the egg does finish meiosis 1, but the major changes at this point is truly within the follicle. And that's why you see all these changes. So you can see from the from the primordial follicle, it becomes the primary follicle. From the primary follicle, it becomes the secondary, the tertiary, and then the mature. Take a look at the cartoon images of the follicle. Okay, so all that pink is the follicle. So this is the follicle right here that surrounds the egg. This is the follicle here that surrounds the egg. And here's another follicle here. What do you notice? What changes do you see from one stage to the next?
It gets thicker.
Yeah, it gets thicker. Absolutely. Okay, what else? What other changes do you see? How does it get thicker? In shape or in layers? Or both?
It's because
Layers.
it's lining, no?
It's actually in both. So the first change that you'll see is in shape. And then the second stage that you'll see is in layers. So it starts off as what again? In the primordial follicle?
Simple squamous.
Yes. So it starts off as simple squamous, but at the release of FSH, it's actually going to develop from simple squamous to simple cuboidal. And that's what distinguishes it apart from the primordial follicle to the primary follicle. It's the shape. It goes from simple squamous to simple cuboidal. And then it's going to develop even further. Now, not only is it simple cuboidal, but now it's what? It's thicker because of what?
It changes to stratified cuboidal.
Exactly. So the stratified cuboidal epithelium, okay, is what lines the secondary follicle. Okay. And then what changes between the secondary follicle to the tertiary follicle? Maybe not the follicle itself, but the space between the follicle and the egg. What does it develop between the secondary and tertiary?
The third one is a mature egg already, right?
What do you mean by, do you define mature egg to me?
Mature follicle?
Okay, you have to be careful because the egg and the follicle are two different things.
Okay.
Yep. So do you see the yellow ball with the blue ball inside? That's the egg. But
Okay.
all of the pink stuff that surrounds it, that's the follicle.
Okay, got it.
So the major difference between the secondary and tertiary, they're both stratified cuboidal, but in the tertiary, you will find the antrum, and that's significant. Sorry. Good thing we're not doing this in person. You'll find the antrum, and that's the fluid-filled space, among other structures like the sulca folliculi and the zona pellucida. From there, it's going to become the mature follicle, and that's when it's ready to rupture. And in other words, release the egg through ovulation. Okay. So these are the changes of the follicle as FSH is being released. Can someone review that for me again? So go through what the changes are. Can one of you go through the changes?
So first we start with the primordial follicle, right? And then we go into the primary follicle, which is lined with simple cuboidal. And then the secondary with stratified cuboidal. And then the third with stratified cuboidal and the antrum. And the antrum was for creating the zona pellucida.
The antrum is actually, so it's going to contain substances that help to produce the estrogen. Yep. Keep going. You're on a good roll.
To produce estrogen, you said, right?
Mm-hmm.
And then from the third, it becomes mature and it ruptures.
Yep.
And then that's when you ovulate, right?
Yep. Perfect. Now, at the very beginning, when FSH is released, what we didn't cover is that there's actually 25 primordial follicles that go through this process. So each month, 25 primordial follicles go through this process. But if the primordial follicle goes through this process, what also goes along with it? What does it surround?
An egg?
Yeah, exactly. So each month, you're not just losing one egg, you're actually losing 20 to 25. So when I say in females, in the oogenesis and follicular genesis process, it's about quality. So of that 20 to 25, they all go through this process. Now, one of those eggs and follicle combination is going to reach a mature follicle state first. So the first one of those 20 to 25 that reach the mature follicle stage is actually going to cause the other ones to shrink and die off. Okay, so it's like a race to see which one matures the fastest, and that one is going to cause the others to stop. So technically, each month you lose about 20 to 25 follicular/egg structures because only one of them is actually ovulated. The best one is ovulated. Okay, how does it do that? Well, this whole process is driven by what hormone?
LH?
Nope. From the primary to the secondary to the tertiary to mature, all of that is driven by what hormone?
Oh, FSH.
FSH. Now, do you remember when I talked about the antrum? So whichever one develops the antrum first to produce the estrogen causes FSH to go down. Okay. So the first one that reaches a mature is going to start to produce estrogen. And the estrogen is going to cause FSH to go down. And if FSH goes down, what happens to the other one that are still in the beginning stages?
They die.
Yeah, they stop and then they die. They shrink and then they just, break down. So the one that becomes the mature the fastest stops the others from growing. Okay. Where it's still growing. And then as FSH goes down, LH goes up. And when LH goes up, guess what LH is going to do? It's going to cause the mature follicle to do what? That's what you're seeing in this image right here. I'm not sure if you can see my arrow.
It turns into the corpus luteum?
Not yet. So LH, elevated levels of LH occur exactly at day 14, and that's when what happens?
Ovulation?
Yes. So that follicle is going to rupture, and it's going to release that egg. And you can see that in this image right here. Okay, so that's what is released at each month. Okay, so each month, FSH starts the whole process. LH recognizes that one has reached mature status, and then it's going to kick that egg out, and then that follicle stays in the ovary. Now that follicle stays in the ovary, but now instead of being called a mature follicle, it's called the corpus luteum. Okay, so it's called the corpus luteum. Now the corpus luteum has a function because the female body knows that, okay, the egg is now expelled and it's either going to be fertilized or not. But either way, each month I'm going to have to prepare in case fertilization occurs. So the corpus luteum releases progesterone. What does progesterone do?
It makes sure that the endometrium continues to grow or thicken in preparation for a pregnancy, just in case.
You're brilliant. That's exactly it. Okay? I couldn't even say it any better. That's exactly what happens. So the corpus luteum produces that progesterone. And just like you said, that progesterone maintains the endometrial lining. Because it's the lining it's thick with blood in preparation for in case pregnancy happens. Okay, but here's the thing: LH, luteinizing hormone, is what keeps the corpus luteum around. As long as the corpus luteum around, it's going to produce progesterone. As long as progesterone is around, it's going to keep that lining thick, right? But what happens is that once that egg is ovulated, what happens to LH? Does LH stay high or does it decrease after
It-
ovulation?
A decrease.
Exactly. It decreases. And you can see that in this chart right here. Okay. Maybe not that chart. Oh, I don't have it. There's a chart that shows you hormonal levels. Okay. So I did not include that in this one. I'll have to next time. Okay. So when LH spikes, ovulation happens. But once ovulation happens, the LH decreases. Now, if LH decreases, what also follows?
We lose the corpus luteum.
Excellent. You lose the corpus luteum. And if you lose the corpus luteum, what stops being produced?
Progesterone.
Exactly. And if progesterone stops being produced, what happens to the lining?
You get your period.
You get your period. Exactly. So that's why when pregnancy does happen, if fertilization happens, that zygote is going to implant itself along the lining, and it's going to produce a hormone called HCG, human cortical gonadal hormone. And HCG replaces luteinizing hormone. So even if luteinizing hormone goes down, as long as that zygote produces the HCG does the same thing that the luteinizing hormone does in maintaining the corpus luteum. And as long as HCG is there, the corpus luteum stays around. And as long as the corpus luteum is around, it's going to produce progesterone, which is going to stay around for 9 months because that's when the baby leaves. Isn't that like, it's so beautiful and cool how like, how everything is coordinated like that. It's like the body just knows exactly what to do, when to do it. Okay? All right, questions? Does that help make a little bit more sense?
Yes. I thought I didn't know this, but apparently I do.
You do. You answered a lot. Okay. But I like yes, go ahead.
Would you be able to go over the zona pellucida?
Yes.
I guess when it appears, I guess, and I guess what it does.
Yeah, absolutely. Excuse me. So the zona pellucida is that gel-like substance that fills the antrum. So the antrum is the space, and that space is filled with the gel, the glycoprotein gel, and it's produced. It starts being produced at the secondary follicle, but it really truly is a distinguishing character in the tertiary follicle. Okay, did that answer your question?
Yeah, it did.
Yeah.
And I guess, is that gel supposed to like protect the follicle or like?
No. Yeah, that's a great question. So that gel is what gets converted to estrogen.
Oh, gotcha. Okay.
Yeah.
Thank you.
Okay. All right. So that. Oh yeah. So you can see over here on the left-hand side where if fertilization happens, then you finish meiosis 2, and then it becomes a zygote and then an embryo. And if fertilization doesn't happen, it just stays as a secondary oocyte, and that's what's released through menstruation. Okay, questions? All right. Oops. Good. Okay. And then we don't have time to go through the menstruation cycle, but this is what it would look like. So you can see that the cycles go by day 1 through day 28. So that just as there's a name for the ovarian cycle with the follicular phase and the luteal phase, there's also stages of the uterine cycle, which includes the menstrual phase, the preovulatory phase, ovulation, and the post-ovulatory phase. There's a table. Let's see here. No, where is it? Yeah, here we go. We'll go here. So there's a table. Did everyone get a chance to look at this table right here?
Yes.
Perfect. If you haven't, make sure you grab it. It's on the website, the navigational materials. So I created this table specifically for this class because it just, you know, it's a nice way of looking at it in terms of like what's going on. You can see both the oogenesis, the follicular genesis, and the uterine cycles on here. And you can see how it's divided into their own phases and what happens exactly at each of those phases. So this is a really good chart to study while you're preparing for the exam. Okay? All right. And that will kind of walk you through what happens to the uterine lining. All right. So with the last 5-ish minutes, I want to go through spermatogenesis so that you can have something to compare it with. Okay. Now, spermatogenesis is much easier than the oogenesis because it's very straightforward. Whatever happened over there, used centuries. The process itself happens over here. It just is a little different. So one main difference is that spermatogenesis occurs when? During the male fetal development, at puberty. When does it start? And when does it end?
To end?
Say that one more time.
To end.
Yep. Okay. So you're right. You identified what the spermatogonium consists of, 2N. Very good. So you're right. It is diploid.
Starts at puberty.
Yes. So spermatogenesis occurs at puberty.
And then it lasts 74 days, right?
That's interesting. So it's not that it lasts 74 days, but it lasts the whole lifetime.
Oh, yeah.
Male's entire lifetime. So, and the reason for that is because they're producing new sperm all the time. Okay. All right. So that's one major difference. And that's why a male that's 100 years old can have a viable child with less chromosomal mistakes versus females. Because females, when she ages, her eggs ages as well. Where in males, you know, they're making new ones consistently. And because of that, their sperm is viable longer. Okay. All right. But the process is very similar. So you have the spermatogonium instead of the oogonium. You have the spermatogonium. It undergoes mitosis. It's going to result in 2 cells. So remember, mitosis is where it does what again?
Copy and divide.
Yes, ma'am. So temporarily from 2N, it's going to go into 4N. And then when it splits and divides, this is what results. A type A spermatogonium and a type B spermatogonium. The type A spermatogonium is going to repeat this process throughout the male's entire life. The type B spermatogonium differentiates and becomes what? What has it decided to become?
The primary spermatocyte.
Yep. So at this point, it's going to undergo. So when it becomes this primary spermatocyte, it's going to undergo meiosis 1. What happens in meiosis 1?
Divide.
Yep. It's going to divide. So it's going to go from 2N to 2 cells that have what each? I heard 1N.
1N each.
Yep. So then that's when it becomes a secondary spermatocyte. Now the secondary spermatocyte is going to complete meiosis 2. And in meiosis 2, what happens here?
Each N divided into 2.
It copies first and then it divides. Okay? So here, temporarily from 1N, it's going to copy to 2N. Same thing here from 1N to 2N. And then it's going to divide and split into these 2 cells here. It's going to split into these 2 cells here. So it's going to make, from 2 secondary spermatocytes, you're going to have 4 spermatids. How many chromosomes will you find in each spermatid?
23.
Yep, 23 or else 1N.
And 92.
Huh? I heard a 92, right?
Yes.
Is there any 92 in here? So at meiosis 2, will you ever have a cell that has 92?
No.
No, because you only had one. After meiosis 1, it split, so there's just 1N in there. So 1N, when it copies, it can only become 2N, and that's 46. Okay. All right. So let's say, for example, that this primary spermatocyte has to have what on the 23rd chromosome? The 23rd chromosome has to have what?
An X or a Y.
An X and a Y. It must have Y. Okay. So when it divides, that means that this one right here is going to have one of them, and this one over here is going to have the other. So what should I write on this first spermatid in terms of the 23rd chromosome?
X.
Yep. The second one?
X.
Third one?
Y.
Fourth one?
Y.
Okay. Now these spermatids are going to mature and become sperm cells, and that's what will be released, ejaculated, and then it can potentially fertilize an egg. So if sperm number 1 right here fertilizes the egg, what are you going to have in terms of gender?
A girl.
Yep. How about this sperm number 3 right here?
A boy.
Yep. Make sense? So in males, it's about quantity. In female, it's about quality. In males, it's about quantity. So that's why in males, you don't have the polar bodies, right? For them, it's about getting that chromosome across and spreading their genetic information on. And that's why the sperm has very limited, it just has the chromosome, what it needs to penetrate the egg, and some energy to help it move. Okay, questions? Isn't this process much simpler in males than in females?
Yes.
Yeah, but this is essentially why in females it has to finish meiosis 2, because this is just in nature. This is the process that has to happen. So if you go through meiosis 1, it must go through meiosis 2, even though technically it didn't really need to, but it has to finish the process. Okay? All right. So here's a comparison between the oogenesis and spermatogenesis. All right. And is there any questions before I move on to doing a practice quiz with you.
Can you give the main things make them different?
Say that one more time.
The main things make them different between the spermatogenesis and oogenesis.
Oh, yeah, that's a nice question. So one major difference is what? How about your class tells me? What's one major difference?
Sperm doesn't have polar bodies.
Yeah, absolutely. What's another way of saying that? Or another way of saying that is that in oogenesis, the end result is one egg that's released per month. Where in spermatogenesis, it's always going to result 4 sperms. Why? Because the egg undergoes polar bodies where the sperm cells do not. Okay, what's another difference?
Women do all the work.
Not true. So, but yes.
LOL.
Well, I'm sorry.
LOL.
Yes. So in females, their main goal is about quality because the body of that egg will become the body of the zygote. Where in males, it's about chromosomes. It's getting that chromosome across. Okay. What's another major difference? When does the process start?
We start in the womb and they start at puberty.
Yep, exactly.
Do they also have FSH and LH and all those hormones as well, or no?
What do you think?
Yes.
They do. Yes, they do. So FSH triggers, remember when we went through the hormones?
Mhm.
So you can see that the follicle stimulating hormone targets sex cells in general. So it
Okay.
targets both the egg and the sperm. And the luteinizing hormone does the same thing. It targets both male and female. Okay. There is one major difference, though, is that instead of estrogen in females, males have what?
Testosterone.
Yeah. Yep. Okay. All right. And okay, so that's a good question. All right. Any other questions before I move on?